Welcome, and thank you for your interest. At NeoImmuneTech, we're expanding the frontier of Immuno-Oncology and beyond. We were founded in 2014 and based in Rockville, Maryland. We are a public company listed on Kosdaq, have a market capitalization of about $1 billion. Our lead asset, NTI-7, is the only long-acting IL-7 in clinical testing, and we believe it's uniquely positioned to address unmet medical need in immuno-oncology and other therapeutic areas. IL-7 is perhaps the most important cytokine for lymphocyte development and survival, can enhance immunity to cancer as well as infectious diseases. In fact, we have seen early clinical efficacy signal in 3 immune cold tumors, MSCRC, pancreatic cancer, and GBM, and we are optimistic in immune hot tumors as well. Our company is led by our scientific founder who invented NTI-7, and the senior management team are complemented by executives with very rich industry experience in commercial strategy, product positioning, and clinical development. In hematopoiesis, IL-7 is the cytokine that commits the precursor to lymphocyte lineage, and then it specifically helps the precursor to develop into T cell and also natural killer cells. IL-7, however, in its natural form is very challenging to administer to patients because of it is a structural instability and also short half-life. NTI-7 is 2 IL-7 molecules fused to high Fc domain, and this allows extended half-life in patient, improving PK/PD profile. In addition, there was N-terminal engineering to allow higher productivity, higher stability and productivity in CMC manufacturing process. NTI-7 has a distinct product profile in IO based on IL-7 biology. Without IL-7 signaling, 99% naive T cells do not survive to maturity. IL-7 is also critical for memory T cell differentiation and survival. It has also been shown IL-7 helps sustain immune response to chronic antigens and promote lymphocyte homing to tumor or infection site. At the so-called homeostatic cytokine, it also has an excellent safety profile, a clear differentiation from other cytokines like IL-2. Based on its biology, IL-7 is an ideal partner for in combination with checkpoint blockade. With a strong enabling technology and intellectual property right protection, NTI-7 could offer NIT and its strategic partners exclusive competitive advantage if proven in clinical testing. In fact, in both MSCRC and pancreatic cancer, where no response expected for pembro alone, 3 out of 17 evaluable MSCRC patients treated with NTI-7 in combination with pembro achieved PR by iRECIST. 1 PR by RECIST 1.1 was observed in 17 evaluable pancreatic cancer patients. It is important to know that in pancreatic cancer patients, the follow-up was too short for IPR in most patients. Responses once occurred are durable and continue to deepen over time. In terms of safety, the combination treatment of NTI-7 and pembrolizumab was well tolerated, with a low number of grade 4 drug-related AE and no grade 5 AEs. Also, favorable PFS and OS was observed in NTI-7-treated high-grade glioma patients after chemoradiation. Here is the waterfall plot for clinical responses in MSCRC patients. Again, among the 17 evaluable patients, the overall response rate was 18% per iRECIST, and the disease control rate was 59% per iRECIST. 1 patient achieved a confirmed PR with 56% tumor reduction at the 3rd follow-up. There were also 2 pseudo-progressions before response was observed. 1 particular patient who developed progressive disease at the first follow-up, but since then had tumor regression, and the tumor regression continued to deepen over time to reach partial response, and eventually the tumor reduction was 58% by the 5th follow-up. This is the swimmer plot. You can see the durability of responses as well as the dynamic of responses. You can see that the response by RECIST 1.1 occurred relatively early at week 12. The two IPR occurred a little bit later at week 18 and week 24, respectively. This is the swimmer plot for pancreatic cancer patient. As you can see, most of the patient has not had follow-up beyond 12 weeks, allow observation of IPR. However, there's one prolonged stable disease in addition to the confirmed durable PR observed. This is the patient who have a confirmed PR. As you can see that the tumor reduction started relatively early as a first follow-up and continued to deepen and eventually reached 72% tumor reduction. In addition to MSCRC patient and pancreatic cancer patient treated in combination with pembrolizumab, in HCG patient, high-grade glioma patient treated with NTI-7 alone also showed favorable PFS and OS, as you can see in here. So what is the science behind the early clinical efficacy signal seen with NTI-7? Well, based on the response dynamics and IL-7 biology, we hypothesize that NTI-7 works by stimulating a subset of T cell to proliferate and attack tumors. In immune cold tumors, the number of this T cell may be too low for PD-1 blockade alone to show clinical responses. However, in presence of NTI-7, this T cell can proliferate to sufficient numbers over time for clinical responses. As a result, once response occurs, it can persist and continue to deepen. In immune hot tumors, where there are more tumor-reactive T cells, NTI-7 can be expected to expand and deepen responses to anti-PD-1 or PD-L1 therapy broadly. And consistent with this scientific hypothesis, we see not only increased T cell infiltration in tumor microenvironment, but also dramatic increase in the so-called stem cell memory CD T cell in the blood. And this dramatic increase of TSCM was observed in patients receiving NTI-7 alone, as well as NTI-7 plus pembro. This slide shows that NTI-7 treated patient has a preferential increase of TSCM. A TSCM, or stem cell memory CD8 positive T cell, has a self-renewal capability and has been hypothesized to be the key subset of T cell responsible for clinical efficacy observed with checkpoint blockade, like PD-1 or PD-L1 therapy. And in here, you can see that adding NTI-7 boosts across the board all T cell subset, but the TSCM is particularly enhanced by like 50-fold versus 5-fold in other T cell subsets. And we believe that this might be the reason behind the efficacy enhancing property of NTI-7 to pembrolizumab. This TSCM enhancing effect is not only observed when NTI-7 was administered in combination with the pembrolizumab, but also when it was used alone in high-grade glioma patients who were treated with chemoradiation therapy. And as you can see, that the TSCM is clearly significantly boosted after administration of NTI-7. Furthermore, NTI-7 enhanced lymphocyte infiltration into MSCRC and also pancreatic cancer patient. And this enhanced lymphocyte infiltration is associated with better clinical outcome in patients treated with NTI-7 in combination with pembrolizumab. So how do we go from here? And we believe that the business opportunity is significant. The immune cold tumor at least have $15 billion per year business opportunity is the holy grail for novel IO mechanism action. It's high risk, high return, but also could potentially to have fast to approval opportunities. In the immune cold tumor, there are 3 of the largest tumor types, including 95% of CRC, most of prostate cancer, and also most of breast cancer. And in here, we have seen promising clinical efficacy in 3 immune cold tumors, MSCRC, pancreatic cancer, and also high-grade glioma. And in MSCRC in particular, we see pretty provocative clinical efficacy. The second business opportunity is PD-1 progressors. And again, this is at least a $15 billion per year business opportunity, and it has a relatively low efficacy bar with limited competition and also faster market possibility. And here we are continue enroll patient with NTI-7 in combination with pembro in CPI-treated non-small cell lung cancer, non-small cell lung cancer, triple-negative breast cancer, ovarian cancer, and also NTI-7 in combination with atezolizumab in checkpoint-treated skin cancer patient. Last but certainly not least, the largest IO business opportunity is the PD-L1 naive patient in the so-called immune hot tumors. This is much bigger business opportunity, but higher efficacy bar to enter and need to compete with multiple PD-1 combos like a checkpoint, VEGF, etc. In here, we are testing in combination with Tezo in first-line non-small cell lung cancer and to pursue a chemo-free regimen potential for first-line non-small cell lung cancer. Beyond combination with checkpoint, NTI-7 can also potentially in combination with CAR-T and TCR-T, and we have ongoing trial to demonstrate clinical proof of concept. Based on science of IO7, NTI-7 could also enhance T-cell engager and combination with cancer vaccines, other cytokines, chemo, radiation, etc. And we plan to expand into this setting down the road. Based on the mechanism of action and biology of IO7, NTI-7 could also play an important role in infectious diseases, particularly when there's no other treatment options. And potentially also, IO7 has also been investigated for sepsis and immune reconstitutions. In summary, NTI-7 is the only long-acting IO7 in clinical testing. It is uniquely positioned to address unmet medical needs in immuno-oncology and other therapeutic areas. In fact, early clinical efficacy signal was seen in 3 immune cold tumors, MSCRC, pancreatic cancer, and GBM. We are strengthening clinical data sets with a longer follow-up on more patients with MSCRC and pancreatic cancer, and we'll continue to investigate in other immune cold and hot tumors, and we are evaluating patient selection strategy to further enrich patients who would respond to the combination of NTI-7 and pembrolizumab. Thank you for your attention.